Welcome to AP Biology, and today we are concluding enzymes. Let's get this done.
All right. So our first discussion, I think, is a very, very important discussion for us to have. And what we're about to discuss, I think, is really relevant for technology and society today. So what we've been talking about is how external factors affect enzymes. We just reviewed that enzymes are affected by temperature changes, they're affected by pH changes. Sometimes, and most times, enzymes require helpers. What did we say those helpers are called? They're called cofactors, right? And there's two types of cofactors. If they are inorganic, like a mineral, like zinc, we call it a cofactor. If it's organic, we call it a coenzyme. And most vitamins are coenzymes, right? What does that mean? It means that these are chemicals that are bound to an enzyme, and that enzyme does not work properly unless it has its cofactor or coenzyme bound to it. Does that make sense to you? Right? And we ended class by saying this is what makes having vitamins in your diet so critical. You are your enzymes. Everything that happens inside of you is a chemical reaction. Every chemical reaction requires an enzyme. So if you have a nutritional deficiency where you are not getting the proper amount of vitamins that you should, that means that your enzymes are not working properly and some metabolic process is not working as efficiently as it should be in your body. Now, I know most of your diets, right? And I know the American diet. I know what most people eat. If you are not taking a multivitamin, do you think you're getting all your required dosages of vitamins? Absolutely not, right? The American diet consists of super highly processed foods that are robbed of all their nutritional value, including their vitamins. Right? So unless you have a diet that's really rich in fruits and vegetables, I doubt you're getting the amount of vitamins that you should. Right? Each of you should be taking a good quality multivitamin that contains vitamins and minerals to aid in the functioning of all of your enzymes. I was looking at Emergency. You ever see that? Yeah. And there's also, what's the other one that you take before you get sick? Emergency is one of the big ones. Airborne. You ever see Airborne?
Yeah.
And they all said they contain zinc. And zinc is supposed to be really good for your immune system. You ever hear that?
I've never heard that.
No. If you're getting sick, you're supposed to take zinc. It's supposed to help your immune system. But now you understand why. Right? Zinc is an important cofactor for many of the enzymes that are used within your immune system. So if those enzymes are missing zinc, that means that your immune system is not working to the full strength of what it should be. Does that make sense? So by taking the zinc, it helps those enzymes work better and will help your immune system to become more efficient. All right. Today, we're going to transition to a different set of chemicals. And these are going to be chemicals that shut down enzymes. We call them inhibitors. There's different types of inhibitors, but we're going to focus on two of the major types. One called a competitive inhibitor and one called a non-competitive inhibitor. Inhibitors are chemicals that bind to enzymes and stop them from working. So are inhibitors necessarily a good thing or a bad thing? They're typically a bad thing, although today we're using them for good purposes. And I'll show you some examples. The first type of inhibitor is called a competitive inhibitor. So take a look at this diagram on the board because it shows the whole system really well. In purple, you see the enzyme, right?
And this region right here, we should all be looking up here. This region right here is the enzyme's active site, right? And that is where the substrate binds to. And we can see the lock and key mechanism right here. You see how they fit together in a very specific way, right? A competitive inhibitor is a chemical that looks like the substrate. So it's able to bind to the active site of the enzyme. And if that inhibitor is bound to the active site, can the substrate get in? No. So it's literally blocking the substrate from being able to get into the active site of that enzyme. So a competitive inhibitor will stop an enzyme from working. Now, something that you should write down, there's a very specific way that you can overcome a competitive inhibitor. Now, the way that I think of it is it's like a war, right? To see who gets into the active site first, the substrate or the competitive inhibitor, right? And whoever beats the other one into it is the one that gets into the active site. So by that logic, how can you overcome the action of a competitive inhibitor? Does anybody know? It's hard to kill something specific. Well, they're both moving faster then, but you're on the right path.
Could you have like 2 spots for just
No, you can't change the spots that things bind to. You can't affect the enzyme. Aaron? Yeah, add more substrate to it. Think about it, right? Think about a war. If you have 2 armies of the same size, right? Then it's kind of like equal. You add more of one type, right? And more likely they're to win. So by adding more substrate, you have a greater likelihood of the substrate going to the active site than the inhibitor. Does that make sense? So there's a word that you should write down. Competitive inhibitors are reversible. They are reversible. You can overcome or you can reverse the action of a competitive inhibitor, and you can do this by adding more substrate. Because if you add more substrate, in a way, it's like the substrate wins the war. Now, it's hard to come up with great examples of competitive inhibitors. So I did some research, and I found out some really cool stuff. I found on the internet this really science-y page. Let's check it out real quick. All right? And this super science-y page talks about competitive inhibitors, right? And today, one of the best things that we are using competitive inhibitors for is to combat cancer. So many of the cancer chemicals that we take, when we are taking, you ever hear the term chemotherapy?
Yeah.
Right? We're going to talk a lot about chemotherapy in the future. But you're taking in a chemical that is targeting and killing cancer cells, right? So many of these chemicals that we are using are competitive inhibitors of enzymes that a cancer cell uses to reproduce. So if we can inhibit an enzyme that a cancer cell uses to reproduce, then that cancer cell can no longer divide, right? And you're killing the cancer. So this is how a lot of new chemotherapeutic drugs are being developed. And it says one right here. An example of a competitive inhibitor is the antineoplastic drug methotrexate, right? Methotrexate has a structure similar to the vitamin folic acid. It acts by inhibiting this enzyme, preventing the regeneration of that. This interferes with DNA synthesis and blocks cell division in rapidly dividing cancer cells, which is the only important thing, right? So this chemical, methotrexate, is a competitive inhibitor of an enzyme that cancer cells use to make their DNA. And by competitively inhibiting this enzyme, you're able to kill rapidly dividing cancer cells. Pretty crazy, right? But it's a use of competitive inhibitor.
Doesn't caffeine do something like that, but kind of the other way around? Like when you drink coffee, the caffeine is, I think it's shaped similar to serotonin.
Yes.
It's like pretending that serotonin is actually there.
It's not serotonin, but so caffeine is not, as far as I know, an inhibitor. But it does mimic. It's more of a mimicker. It resembles other chemicals in the body, right? And it binds to their receptors. But it's not, as far as I know, I don't think it's inhibiting anything. But you're right. In a way, it's working like a competitive inhibitor where it mimics a specific chemical in the body. All right? Well, I'll look that up because that's really interesting. Or you should look it up when you have a chance to tell me the answer. Right? Jeff?
Has this been recently discovered? Or?
I don't know. It's a good question.
Because
Right?
if so, this is
not a typical chemotherapeutic drug. The most common one is one called Taxol. But Taxol works in a very similar way.
If this is the case, then cancer could be in the end game right now.
But no. So here's the thing. And
I'm serious.
no. So here's the thing. When scientists are looking for medicines and ways to treat specific diseases or cancers, when they are searching for a drug to be used, right? Many drugs work as inhibitors. Many drugs do. I'm going to give you even more examples of that, right? But this is an example of a drug that works specifically as a competitive inhibitor. And the thing that I really want you to know about a competitive inhibitor right now is that it's reversible, right? It's reversible. It doesn't work forever. Does that make sense to all of you guys? Now, the other type of inhibitor is called a non-competitive inhibitor. And to me, these guys are much more widespread, and they're much more harmful than a competitive inhibitor. So look at what a non-competitive inhibitor does. It doesn't bind to the active site. Instead, it binds to a different region, and it changes the shape of the active site. Now, if you change the shape of the active site, can that substrate bind any longer? No, absolutely not. And that enzyme is functionally shut down. Now, here's the thing about a non-competitive inhibitor. They are irreversible. They are irreversible. You can add as much substrate as you want. Is it going to do anything? No. It doesn't matter how much substrate there is if there is no active site for them to bind to. So non-competitive inhibitors, in essence, kill the enzyme, right? A competitive inhibitor is blocking it. A non-competitive inhibitor is killing it because it's irreversible, right? It's going to have much more harmful consequences to the actual organism. These guys are super relevant to biotech today. For example, antibiotics are all non-competitive inhibitors. That is how they work. So if you think of typical antibiotics, what are some examples of antibiotics? Penicillin, ampicillin, amoxicillin, tetracycline, you name it. All these chemicals are non-competitive inhibitors of enzymes that bacteria need to reproduce. If you look at a common one like penicillin, penicillin is a non-competitive inhibitor that a bacteria uses to make its cell wall. The bacteria can't make its cell wall, what happens to it? It dies. Right? And that is how antibiotics work. That is how antibiotics kill bacteria. Right? They bind to and they turn off key enzymes that bacteria need to reproduce.
Now, can antibiotics differentiate between your good bacteria and your bad bacteria? No, they kill all the bacteria in your body. They can't differentiate. They're non-competitive. Non-competitive. They are irreversible. They kill all that bacteria, and that's it. If we hear about somebody being poisoned, a toxin, toxins are non-competitive inhibitors. Have you ever heard of a gas called nerve gas?
Yeah.
Right? It's a gas, it's called sarin. In the 90s, I believe, it was released in a subway in Tokyo, right, as a terrorist attack.
Yeah.
And it is a non-competitive inhibitor of enzymes in the nervous system. So they die a really slow, painful death because their nervous system is no longer working. They lose the ability to breathe and do all that stuff that your nervous system controls. Non-competitive inhibitor, bad stuff. Toxins, poisons, non-competitive inhibitors. Also, pesticides. Pesticides are non-competitive inhibitors. We all know what a pesticide is, right?
Yes.
It's a chemical like DDT, right, that is spread on plants that kills all the insects that would be eating that crop, right? Well, this is how this chemical kills all the pests that would be eating the fruit and vegetables, right? These chemicals are non-competitive inhibitors that these insects need, right? Doesn't make sense. These are non-competitive inhibitors of enzymes that insects need to survive. And by killing those enzymes, you're killing the insects. What's funny is then we then ingest those pesticides and we eat the plant, right? Which is not good for us. Cool, go ahead.
With the enzyme, like the picture of the non-competitive inhibitors, there's like a little slot that was for the non-competitive inhibitor.
Yeah.
Isn't there like, or wouldn't it make sense for the enzyme to adapt by like not having that area for the non-competitive?
Yeah, but enzymes don't just adapt. It's not like a thing. You know, like an enzyme can't just change itself on demand. Right? Evolution doesn't work that way. So it would be nice. You know, it would be possible that a person can be randomly born with a mutation where that enzyme has a different shape where that inhibitor can't bind to it, right? In that case, that person would be naturally immune to that poison. And then could pass down that mutation to their offspring, and then their offspring would be immune to that poison. Does that make sense to you? Yeah. But it's not like the enzyme can just change on demand to compensate for that inhibitor. Good question. Joe, what's your question?
I was just gonna say that I remembered seeing something from a National Geographic back in the day telling people not to spray pesticides over their kids' beds. I'm not even kidding. Like back in the 50s, like people used to spray pesticides in people's rooms.
For like bed bugs and stuff.
Basically, like so many kids were like getting sick and dying.
Yeah, well, pesticides. Yeah, well, yeah. You know, we know today that pesticides are known carcinogens that lead directly to cancer.
Yeah.
All right.
Oh.
But we're not talking about that right now. Right now, what we're really focusing on is what they are, right? And when we look at chemicals that are being used today, whether they are being used for a medicine to treat cancer, whether they're being used as a medicine to kill bacteria, whether they are being used as a pesticide, whether they are being used as a poison. So many of these chemicals that we're using today within biotech are going to be inhibitors, either competitive or non-competitive inhibitors, right? And it should make sense to you because enzymes run the show. If you look at any organism on this planet, whether it's a person or a bacteria, all they are is a little vat of chemical reactions, and every chemical reaction needs a what? An enzyme. So if you target that chemical reaction, if you target that enzyme, you can shut down that chemical reaction and affect that organism. Does that make sense to all of you? Leanna, what's up?
You said like enzymes don't adapt, but why is it that like if someone like injects themselves like little amounts of poison or toxin, like
That's different. That's tolerance.
That's different. Okay.
Tolerance works different than adapt.
Okay. So it doesn't work with enzymes.
No. We'll talk about tolerance later on, right? But that's a good question. Tolerance is different. All right. All right. Are we all good with competitive, non-competitive inhibitors? All right. The last thing that we have to talk about in this chapter is how we regulate our metabolic pathways, right? Think about it. If we were not able to regulate our metabolic pathways, we would have chemical chaos occurring within our bodies, right? So we need to have very, very tight control over our metabolism. We need to be able to turn pathways on or turn pathways off, depending upon whether or not we want those pathways running. So how do we do this? How do we have the ability to turn pathways on, turn pathways off, depending upon whether or not we need them running? Well, we're going to use a very specific type of enzyme called an allosteric enzyme. The process of regulating our pathways through allosteric enzymes is referred to as allosteric regulation. Now, these enzymes, allosteric enzymes, are very special enzymes. They are special because they are enzymes that have the unique ability to be turned on or turned off. So these enzymes have two different formations. They have an active form and they have an inactive form. So it's a little confusing, so take a second, look at this picture on the board. All right. This and this are the same enzyme. Now check this out. These allosteric enzymes are unique. If you look at this allosteric enzyme right here, this is one enzyme, right? Can anybody tell me how many polypeptide subunits it's composed of? It's composed of 4 polypeptide subunits. You see that? And each polypeptide subunit has its own active site. So this allosteric enzyme actually has 4 different active sites. Now, what's unique about this allosteric enzyme is that it has the ability to change shape and turn on or turn off. A chemical called an activator has the ability to bind to the allosteric enzyme and turn it on. And I think this picture shows it really well. See how the activator kind of works like a wedge? Do we all see that? And it kind of sticks in the enzyme and it locks it into its active form. A chemical called an inhibitor can bind to the same allosteric enzyme and essentially turn it off. If you look at this picture, see how the inhibitor is changing the shape of the active sites to turn off that enzyme? Now this inhibitor, this is an allosteric inhibitor. This is not competitive, it's not non-competitive, it's none of that. It's its own separate category. You have an allosteric activator, you have an allosteric inhibitor. Used to turn the enzyme on, used to turn the enzyme off. So these allosteric enzymes are very unique because they are the only type of enzyme that has the ability to be turned on or turned off, essentially on demand. Does that make sense to you? All right. Now, this is used to regulate our pathways, right? To regulate our pathways. Here's another good example, right? So here's an activator binding to the enzyme, locking it into its active form. Here's an allosteric inhibitor binding to its site, turning off the enzyme. Really good picture demonstrating how it works. All right, I'm going to jump ahead a little bit. All right. Let's say you have a metabolic pathway, like the pathway on the board. Don't write anything down for a minute. Just look at the board because this could be really confusing. here is a metabolic pathway. You begin with the initial substrate called threonine, and you end with the end product called isoleucine, right? And this metabolic pathway has 5 different chemical reactions in the pathway. So you have 5 different reactions. How many enzymes do you need? 5. 5 reactions, therefore you have 5 different enzymes, right? Now, if you want to be able to control this pathway, if you want to turn this pathway on or turn this pathway off, which of those 5 enzymes would you make an allosteric enzyme? The very first one. That should make sense, right? You might as well turn the pathway off at the beginning. So usually when you look at a metabolic pathway, the very first enzyme in that pathway is usually your allosteric enzyme. It is your enzyme that can be turned on or turned off. Now here's the thing. Again, looking at this pathway. This pathway is anabolic. It's consuming energy to build isoleucine. When would you not want this pathway running?
I'm trying to conserve energy.
Well, you're trying to conserve energy, yeah, but think about it. This pathway is consuming energy to build isoleucine. Yeah, when you don't need any more isoleucine, right? So when you've made enough isoleucine that you're good, would you want to have this pathway still on making more? No. So when you have enough isoleucine, what do you want to do to this pathway? Turn it off. Now, here's the ingenious part. Isoleucine itself works as the allosteric inhibitor. When isoleucine builds up, it is going to bind to that very first enzyme, that very first allosteric enzyme, and turn off the pathway. Pretty ingenious. So when you have a buildup of isoleucine and you don't need any more, isoleucine itself works as the allosteric inhibitor, blocking that first enzyme, shutting it off. When will that enzyme turn back on? When you run out of isoleucine, right? When you run out of isoleucine, now you don't have any more inhibitor. And now this enzyme turns back on and the metabolic pathway turns back on. So we give this a very specific name. When the end product turns off a metabolic pathway, we call this feedback inhibition. Feedback inhibition is the process where the buildup of the end product shuts down the entire metabolic pathway. Yeah.
Is, like, if we have one of these systems, would all of this be going on in one cell? Or,
Yeah. There's a whole lot going on inside your cells. Now, we're in really good shape. We should be able to wrap up everything within like 10 minutes on Monday. We're in really good shape. Do we feel pretty clear everything today? This end got a little hairy. I'll review this on Monday. We only have a few minutes left. I want to work on a question together. And then we'll wrap all of this up on Monday. All right. You guys can put away all your stuff.